Welcome to our presentation on milk hygiene and public health, presented by Dr. Asmita Bansod from GNP Sir Classes. Today, we will explore the critical aspects of milk hygiene and its impact on public health. Milk, as defined by the Prevention of Food Adulteration Act, is the normal secretion from milking animals. Milk hygiene involves ensuring milk is free from bacteria and other contaminants. Veterinary public health focuses on the health activities related to milk production and safety. Microflora in milk and milk products include various bacteria, yeasts, molds, and viruses. These microorganisms can affect the quality and safety of milk. Bacteria in milk are classified based on their growth temperature: mesophilic, psychrotrophic, thermophilic, and thermoduric. Each type has different implications for milk safety and quality. Key points include the effects of different bacteria on milk, such as Serratia marchescens causing red pigmentation and Clostridium species causing various spoilage conditions. Spoilage in milk and milk products can occur through various processes like souring, gas production, lipolysis, and proteolytic activity, each caused by different microorganisms. Sweet curdling, ropiness, and other spoilage conditions can affect milk's viscosity and texture, often caused by specific bacteria and enzymes. Flavor-producing bacteria can cause fruity, malty, fishy, or unclean flavors in milk. Color fermentations can result in yellow, blue, green, black, or red discolorations. Yeasts like Saccharomyces cerevisiae and Candida species are used in brewing and dairy production, affecting the flavor and texture of milk products. Molds such as Penicillium and Aspergillus species are involved in cheese production and can affect the flavor and safety of dairy products. Bacteriophages can cause slow starter action in cheese production, leading to failures. They enter through raw milk, tools, and personnel, affecting the fermentation process. Raw milk contamination can occur from various sources, including milkers, the environment, equipment, and during processing and transport. Proper hygiene practices are essential to minimize contamination. Mastitis is the inflammation of the udder, classified based on symptoms and causative microorganisms. It significantly affects milk quality and production. Various microorganisms are associated with mastitis, including Staphylococcus, Streptococcus, and Mycoplasma species, each contributing to the disease's complexity. The somatic cell count in milk is a key indicator of its quality. Normal milk has a lower count, while abnormal milk can have significantly higher counts, indicating issues like mastitis. Comparing the constituents of normal milk and mastitis milk reveals significant differences in fat, protein, lactose, and other components, affecting milk quality. Detecting mastitis in milk involves checking pH, chloride content, catalase test, and somatic cell count. These tests help identify the presence and severity of mastitis. Tests like the sodium lauryl sulfate test and direct leukocyte count help determine the severity of mastitis by measuring somatic cells and other indicators in milk. Identifying mastitis-causing microorganisms involves observing hemolysis patterns and using CAMP tests to differentiate between various bacteria. Direct microscopic count (DMC) and standard plate count (SPC) are methods to assess milk quality, with different ranges indicating the milk's condition from good to very poor. Ensuring the production of hygienic milk involves several critical steps. These include cleaning and sanitizing dairy equipment and utensils, maintaining milkers' hygiene, and using the most hygienic milking methods, such as full hand milking. Teats should be washed with a cloth dipped in KM04 or bleaching powder. Adopting clean in place (CIP) methods and other hygienic milking practices are also essential. Quality control tests are essential for ensuring milk safety and quality. These tests include acidity, alcohol test, clot on boiling, dye reduction tests, and many more. Each test has a specific purpose, such as determining bacterial contamination, fat content, or the presence of adulterants. Several important acts and rules govern the dairy industry. These include the Prevention of Food Adulteration Act, Agricultural Produce Act, Milk and Milk Products Order, Standards of Weights and Measures Act, and the Export Quality Control and Inspection Act. Milk can be a carrier of various pathogens, including bacteria like Bacillus cereus, viruses like the polio virus, and fungi like Penicillium spp. These pathogens can cause serious health issues if not properly managed. Milk can undergo various flavor changes due to different factors. For instance, proteolysis can cause a bitter flavor, while aerogenes can lead to a medicinal flavor. Other changes include musty, fruity, phenolic, and fishy odors caused by different bacteria and chemical reactions. Milk-borne poisoning can occur through various means. Infections can be caused by viable organisms, intoxications by ingestion of preformed toxins, and toxi-infections by toxins produced in the body. Examples include salmonella poisoning, staphylococcal poisoning, and bacillus cereus poisoning. Brucellosis is a classic example of a milk-borne disease. Botulism is characterized by double vision and respiratory failure, while bacillus cereus poisoning is known for causing vomiting and the fried rice syndrome. Different species produce milk with varying constituents. For example, cow milk contains about 86.6% water and 4.6% fat, while mare milk has 89.1% water and 1% ash. These variations affect the nutritional value and uses of the milk. The composition of different types of milk varies. For instance, skim milk has less than 0.5% fat, while buffalo milk contains about 6% fat. These differences are important for dietary choices and processing methods. Pasteurization is a crucial process for making milk safe to consume. Methods include high temperature short time, HTST, low temperature long time, LTLT, and ultra-high temperature, UHT, pasteurization. Each method has specific temperature and time requirements to effectively kill pathogens. Detecting adulterants in milk requires specific chemicals. For example, ammonium sulfate can be detected using 2% NaOH and phenol, while glucose can be identified with Barfoed's reagent. These tests ensure milk purity and safety. There are specific tests to detect various adulterants in milk. These include the resorcinol test for cane sugar, the iodine solution test for starch, and the phosphate test for pasteurization adequacy. Each test targets a specific adulterant or quality parameter.